Hello guys, welcome to my channel. Today we'll be doing the part 2 of the biotechnology, its principle and processes chapter. In the previous video, I have already taught you the first part. We have already done with the NCERT. Now we'll be doing the second part. And after that, I'll be sharing some notes that link I'll share in the description box. So maybe in the next video, not in this video. So without wasting our time, let's begin. So now we'll do the part 2 of this chapter, that is the process of recombinant DNA technologies. So recombinant DNA technologies, they involve several steps in specific sequence such as isolation of DNA, then there is fragmentation of DNA by restriction endonucleases or restriction enzymes, then the isolation of desired DNA fragment. Before that we do the gel electrophoresis to separate the fragments, then there is isolation of desired DNA fragment, then ligation of DNA fragment into a vector and formation of recombinant DNA, and then transferring the recombinant DNA into the host using the microinjection animal cell, biolistics or gene gun in plant cells, and the disarmed pathogens in animals as well as plant. Culturing the host cells in a medium at large scale and extraction of the desired product is the next step. First is isolation of genetic material, that is nucleic acid is the genetic material in all the organism without exception. In majority of organism it is DNA. In some or viruses it is RNA. In order to cut the DNA with restriction enzyme, it needs to be in pure form, free from other macromolecules. Since DNA is enclosed within the membranes, we have to break the cell open. DNA जो cell के अंदर है, we have to first cut the cell or break the cell open so that the DNA comes out, and then the DNA along with other macromolecules such as RNA, protein, polysaccharide, and also lipid comes out, and this can be achieved by treating the bacterial cell. In case of bacterial cell, we use lysozyme enzyme to break the cell open. In case of plants, we use cellulase because plant cell wall is made up of cellulose. So we have to use cellulase enzyme to break the cell open. In case of animals, they don't have cell wall, so uh, they have not mentioned the enzyme here. But in case of fungus, we use chitinase because fungal cell wall is made up of chitin. So to digest that chitin, we use chitinase to break open the cell. Finally, after breaking open, macromolecules which come out are DNA, RNA, proteins, lipids, carbohydrates, and many other things. So you know that the genes are located on long molecules of DNA intertwined with proteins such as histones. This RNA can be removed by treatment with the ribonucleases, also called as RNases. They digest ribonucleic acid, that is RNA. Then proteins are removed by treating with proteases, which digest a protein. And other molecules can be removed by appropriate treatment. And now we are left only with DNA. So this purified DNA ultimately is precipitated out using the chilled ethanol मतलब बहुत ठंडा ethanol, that is the alcohol. We use chilled ethanol to precipitate out the DNA. The purified DNA ultimately precipitate out after addition of the chilled ethanol. So यहाँ डायग्राम में उन्होंने दिया है this is the purified DNA which is then precipitated out using the chilled ethanol. It is then removed by spooling process. Here they have given under figure 11.5. Now the cutting of DNA at specific location. This was possible only because of restriction endonucleases or the molecular scissors. The restriction enzyme digestions are performed by incubating purified DNA molecule with the restriction enzyme at optimal condition for that specific enzyme. सभी जो एंजाइम्स होते हैं उनको ऑप्टिमम टेम्परेचर एंड pH चाहिए होता है फॉर देयर हाईएस्ट एक्टिविटी, so that they show their highest activity. Even for salivary amylase, you must have performed enzyme and pH, optimum pH experiments. So similarly, this also enzyme requires the optimal conditions for that specific enzyme. Agarose gel electrophoresis is carried out for the separation of the DNA fragments which are generated by the restriction endonuclease. Then DNA is negatively charged, so it is forced to move towards the positively charged anode through the matrix or the agarose gel, which is the most commonly used matrix. And then after that, so they basically move due to the sieving effect of the gel according to their sizes. The smaller the fragment, farther it will move. Okay, so this process is repeated with the vector DNA also. Now the joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA with a specific restriction enzyme, the cut out gene of interest from the source DNA and the vector DNA with the space are mixed. So you have to use the same restriction enzyme to cut the vector as well as the source DNA. Then only you will get the desired product. So after that, the cutout gene of interest is mixed with the and the ligase is added along with vector. This results in the preparation of recombinant DNA. Now amplification of the gene of interest using PCR. PCR stands for polymerase chain reaction. You must have heard about the RT-PCR during the COVID. So this is similar kind of thing. That is PCR, polymerase chain reaction. In this reaction, multiple copies of gene or DNA of interest is synthesized in vitro. In vitro matlab in the laboratory outside the body is called in vitro using two sets of primers. Primers are small chemically synthesized oligonucleotides that are complementary to the regions of DNA. So we use after uh, after after isolation of those fragments, they are we take them for the PCR. In this reaction, multiple copies of gene or DNA of interest is synthesized in vitro using two sets of primers. Then the DNA dependent DNA polymerase enzyme, and the enzyme extends the primers using the nucleotides provided in the reaction and the genomic DNA as a template. So there are basically three steps in this process. The first step is denaturation, मतलब जो डबल स्ट्रैंडेड डीएनए है उसको आपको सेपरेट करना है बोथ स्ट्रैंड्स को जो फ्रैगमेंट्स बनाए हैं. दिस वेट आई विल शो यू. This whole is a one fragment generated by the restriction endonuclease enzyme. So you have to separate these fragments into two strands. So for that you need to do the denaturation process. After denaturation is carried out using heat. आपको हीट करना है at around 94 degrees Celsius. Then after denaturation there is annealing. In annealing we use primers. Primers are those small chemically synthesized DNA parts. So this is a primer, small sequences. We use two sets of primers here. Then so first is denaturation, then annealing, and then the last step is extension. In extension we use DNA dependent DNA polymerase enzyme which extends the primers and the new strand is generated or synthesized. Here we use special designed DNA dependent DNA polymerase enzyme, also called as the Tech polymerase. So we'll discuss this now. So if the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately 1 billion times. That is around 1 billion times. That is 1 billion copies are made if you repeat these amplifications for many times. You can actually make 1 billion copies, that is 1000 million copies. And such repeated amplifications is achieved by use of special thermostable DNA dependent DNA polymerase enzyme called Tech polymerase, isolated from the bacterium Thermus aquaticus. That is a highly thermostable bacteria which remain active. This bacterium Thermus aquaticus remain active even during high temperature, and that's why we use this thermostable DNA dependent DNA polymerase isolated from this bacterium thermostable bacterium Thermus aquaticus, which can live even at high temperature. And now the amplified fragment is if desired can now be used to ligate a vector for further cloning. अगर आपको अभी वो क्लोनिंग करने के बाद चाहिए to ligate it with the vector for further processing or further cloning and formation of recombinant, you can use it now. And the DNA dependent DNA polymerase isolated from this Thermus aquaticus is also known as the Tech polymerase. Now insertion of recombinant DNA into host or cell or organism, this we have already seen. We have to first make the bacterial cell competent that is treating with divalent cation like calcium. Further, then the efficiency of uptake of DNA increases. Then you have to incubate on ice, followed by keeping them at very high temperature, heat shock at 42 degrees Celsius, and then again followed by ice. This is in case of bacteria. For animals, we can use directly micro injection, in which the recombinant DNA is directly injected into the nucleus of the animal cell. Then for plants, the cells are bombarded with high velocity microparticles of gold or tungsten coated with the recombinant DNA. And this method is called as a biolistics or the gene gun. And finally, we can also use the disarmed pathogens like retroviruses in case of animals, and Agrobacterium tumefaciens, which is a pathogen in dicot plants in case of plants. The ampicillin resistance, okay, so this we have already seen. Now obtaining the foreign gene product. So हमने रिकॉम्बिनेंट डीएनए तो बना लिया और ऑर्गेनिज्म में डाल भी दिया. अभी हमें चाहिए डिजायर्ड प्रोडक्ट, दैट इज़ द प्रोटीन. When you insert a piece of alien DNA into a cloning vector and transfer it into a bacterial, plant or animal cell, the alien DNA gets multiplied. And in almost all the recombinant DNA technologies, the ultimate aim is to produce a desirable protein. Okay. Hence, there is a need for recombinant DNA to be expressed. The foreign gene gets expressed under appropriate condition. The expression of foreign gene in the host cell involves the understanding many technical details. After having cloned the gene of interest and having optimized the condition to induce the expression of the target protein, one is to consider producing it on a large scale. So if protein encoding gene is expressed in a heterologous host, it is called as a recombinant protein. And these cells harboring cloned gene of interest cells may be grown on a small scale in laboratory. The cultures may be used for extracting the desired protein and then purifying it by using different separation techniques. Okay. So now the small cultures, the small volume cultures, they cannot yield appreciable quantities of products. To produce in large quantities, development of bioreactors, also called as the fermenters, where large volumes around 100 to 1000 liters of culture can be processed, was required. And thus a bioreactor can be thought of a vessel in which raw materials are biologically converted into a specific product or individual enzyme, etc. A bioreactor provides the optimal conditions for achieving the desired product by providing optimum growth conditions like temperature, pH, substrate, salts, vitamins, oxygens, etc. So here two different types of bioreactors are shown. The first one is the figure A shows the stirring type bioreactor, and B is sparged stir tank bioreactor. The most commonly is the A one, that is stirring type bioreactor. A stirrer tank reactor is usually cylindrical or with a curved base to facilitate the mixture or the mixing of the reactor contents. The stirrer facilitates even the mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. If you look at the figure closely, you will see that the bioreactor has an agitator system, an oxygen delivery system of just mark this. This is very important. A bioreactor, the stirrer facilitates oxygen and mixing. It facilitates mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor, and at the same time, it has agitator system, and oxygen delivery system, foam control system, and temperature control system, pH control system, as well as sampling ports, so that the small quantities of culture can be withdrawn periodically. Now downstream processing. Downstream processing is nothing but after completion of biosynthetic phase, that is production of this protein, the product has to be subjected through a series of processes before it is ready for marketing and finished. The process includes separation and purification, which are collectively referred to as downstream processing, and the product has to be formulated with suitable preservatives. Such formulations has to undergo thorough clinical trials as in case of drugs. Strict quality control testing for each product is also required. The downstream processing and quality control testing vary from product to product. So we are done with our biotechnology principles and processes chapter 1, and this is the summary. If you want, you can go for this and please solve this exercise. Just look at these questions, they might be helpful for the boards as well as for NEET.